going to talk a little bit about the Krebs cycle. I think for the Krebs cycle, the actual products are a little bit more important to know than the actual enzymes. And it's just important conceptually to know what the Krebs cycle is, what its products are, and basically what it's used for. So the Krebs cycle is also known as the citric acid cycle, the tricarboxylic acid cycle, TCA cycle for short. It takes place in the mitochondrial matrix. So remember, from glycolysis, we're coming over from the cytoplasm to the PDC complex is by the mitochondrial matrix, and so is the Krebs cycle. It's inside the mitochondrial matrix. So acetyl-CoA is the product of the pyruvate dehydrogenase complex that we were just talking about. Pyruvate goes in, acetyl-CoA comes out. This acetyl-CoA can now spin the Krebs cycle. So it enters and it combines in the first step with oxaloacetate to form citrate. So I think I go in depth. Yeah. So we're going to talk about all of this. So if it seems a little bit overwhelming, don't worry. I'm just super quickly summarizing it. It combines with the oxaloacetate that's already in the cycle. This, if you look to the right, this acetyl-CoA is coming in new, but this oxaloacetate is already in the cycle. So it combines, and the first step produces citrate. But basically, that's basically what the cycle is. They combine, they form citrate, then it goes through a series of steps where it's oxidized, and it goes back to oxaloacetate, and then it combines again with the next acetyl-CoA that comes in, and the cycle spins round and round. The Krebs cycle produces 6 NADH and 2 FADH2 and 2 GTP molecules per every original glucose. So remember, when I say original glucose, it's per 2 spins because there are 2 acetyl-CoA molecules that came from that original glucose. So the glycolytic pathway has 10 steps. The Krebs cycle has 8 steps. So let's get into it. So the Krebs cycle, step 1, we have acetyl-CoA coming from the pyruvate dehydrogenase complex. It enters the cycle. It combines with oxaloacetate, and it forms citrate. Sometimes you'll see it as citric acid. I do believe at physiological pH, it's just citrate though. And the enzyme, I did put the enzymes on here just for your information. If you want to try to memorize them, you can. Enzyme for the first one is citrate synthase, and some of them, obviously the names are a little bit easier to intuit where they fit into the cycle, and some of them aren't. So like the enzyme for step 1, citrate synthase, synthesizing citrate. That's pretty obvious. Step 2, aconitase, you might not know it unless you know the Krebs cycle. But like I said, we should focus more on the actual products than the enzymes for the Krebs cycle. So the citrate in step 2 is converted into its isomer, isocitrate. We're just moving it around. That's really it. Step 3, isocitrate is oxidized into alpha-ketoglutarate, which is a 5-carbon chain. And alpha-ketoglutarate is a pretty important molecule. It's an intermediate for the Krebs cycle. I think it's also used as a precursor for amino acids. So it's used in a couple of parts of your body. So it's an important thing to understand that all of these reactions that are happening, including glycolysis and the Krebs cycle and any sort of reaction that happens in your cell, they're happening in tandem with a bunch of other reactions that are occurring around them. So this is in vivo, which means it's inside of a living system. So there are going to be a bunch of other reactions that are happening all at the same time. So just because we're talking about something moving through the Krebs cycle, the next step, it turns into alpha-ketoglutarate, and then after that, it turns into succinyl-CoA. That doesn't mean that the alpha-ketoglutarate that was produced isn't going to go to another reaction. This is just, we're following it because we're following the Krebs cycle right now. But step 3, it's oxidized into the alpha-ketoglutarate, and it also produces a CO2 and an NADH. And the enzyme that is responsible for this reaction, or that catalyzes this reaction, is known as isocitrate dehydrogenase. Okay, so we now have our alpha-ketoglutarate. We let go of a carbon dioxide molecule, and we're going to move on to step 4. So it is now decarboxylated. Remember, that means we're taking a CO2, a carboxyl group, and we are releasing it again as a CO2 molecule. And it is a 4-carbon molecule now, and it's going to be combined with coenzyme A, and the product after step 4 is going to be succinyl-CoA. So with another CO2 and NADH form, just like step 3. And the enzyme that is used is alpha-ketoglutarate dehydrogenase, because we are dehydrating again, and it's a decarboxylation. So we release that carbon dioxide, and now we only have 4 carbons. So we're right over here. We now have succinyl-CoA over here, and then step 5, it is being converted into succinate. And here, the GTP and 1-succinate are produced. So it is a GTP that's produced. And the enzyme that facilitates that is succinyl-CoA synthetase. And it's important to know that the synthetase in this reaction is not actually synthesizing. It's doing the exact opposite. It's breaking down. So we now have succinate over here. So step 6, succinate is converted into fumarate as 2 hydrogens are transferred to an FAD. So we've been dealing pretty much with NAD for the Krebs cycle, NAD+. FAD is also that oxidized form of the high energy electron carrier. So after step 6, succinate is converted into 1 fumarate molecule, and we also now have 1 FADH2 molecule. So that is also going to head over to the electron transport chain, and it's going to facilitate with ATP synthase. And the enzyme here is succinate dehydrogenase, which also is a complex in the electron transport chain. But I'm sure we'll get more into that when we do that lecture. So we're right over here. We have fumarate. So now step 7, we're just adding a hydroxyl group to that fumarate, and it produces malate. And the enzyme that we use is malase. So you can see that all of these steps, we're basically just making little modifications to each of these molecules. Sometimes we'll cleave them, sometimes we'll add them, sometimes we'll just take off a functional group. And every time we do that, we're releasing a little bit energy, or we're transferring a little bit energy from, say, step 8. Say we have malate going to oxaloacetate. We're transferring a little bit of that energy to the NADH, to the NAD+ to create NADH. And then oxaloacetate is a little bit lower energy than malate. So that's where the energy is going. It's all about the energy transfer. And that step 8 is actually what regenerates the oxaloacetate, and that's what keeps the cycle going. So we started with acetyl-CoA and a combination of oxaloacetate, and we're basically just oxidizing it all the way through the entire cycle until we finally get acetate again. And then once the oxaloacetate, excuse me, and once it is able to combine with another acetyl-CoA molecule, we're just going to keep on doing it over and over again. And it's very beneficial. It makes a lot of those high energy electron carriers, and it makes some GTP also. Yeah. So the Krebs cycle creates FADH2 and NADH for the electron transport chain, but it's also responsible for supplying other biosynthetic reactions with reactants. So like we talked about before, alpha-ketoglutarate may leave the cycle to be converted into amino acids. And the cycle is regulated by many factors. So obviously when you have this many moving parts, you're going to have a lot of different places where we can inhibit and where we can activate. So a couple over here, products of the cycle like NADH are going to directly inhibit enzymes in the cycle. So it's just that same exact thing that we always see, where the products of a reaction are going to inhibit the enzymes of that reaction and slow it down. So when we have too much NADH, when we have a lot of these high energy electron carriers, the cell realizes that it doesn't really need to make them, and it can conserve the energy and it can use it for other things. So it's going to inhibit the cycle. It's going to inhibit the enzymes directly. Same thing, citrate inhibits PFK1 activity in glycolysis. Because the citrate is going to be a product of the acetyl-CoA, which is a product of glycolysis. So PFK is the committed step, so it's a pretty good place to regulate. It's a pretty good place to regulate the pathway. So if we have too much citrate, that means that we have enough. We don't really need any more materials to spin the Krebs cycle. So we're going to downregulate that PFK activity so that anything that's coming before, say we have that G6P, we can, shuttle that off to glycogen, and we can add it to our glycogen stores instead of making acetyl-CoA and spinning the Krebs cycle for energy that we don't need. So your body is a very fine-tuned survival machine, and it's very sensitive, these sort of regulations. And calcium accelerates the link reaction. Link reaction is just another way to say the activity of the pyruvate dehydrogenase complex, and it upregulates the Krebs cycle. So if we have a high calcium concentration, we're actually going to upregulate the Krebs cycle. because the pyruvate dehydrogenase complex is sensitive to calcium. Cool. So 100% of you said D, and that is absolutely false. True. So which statement is false? It's also known as the tricarboxylic acid cycle. That is true. It occurs in the mitochondrial matrix. That is true. It spins twice for every starting glucose molecule. That is true. And oxygen is required. That is not true. Oxygen is required in the ETC. It's the final electron acceptor, but the Krebs cycle does not require oxygen. We're just spinning acetyl-CoA and we're just modifying it. The citric acid.